Hi everyone, welcome to the Molecular, Cellular, and Integrative Biosciences Recruitment Research Talks. To give you a sense of what it's like to be a graduate student at Penn State, we've put together a series of short presentations highlighting research interests of MCIBS students and the resources that support them. So I'll kick this off by telling you about my research. My name is Laurel C. Miller. I'm a fourth year student in the MCIBS program. I have an emphasis in neurobiology. My advisor is Tom Gould, and our lab is located in the Biobehavioral Health Department at Penn State. So our lab is really broadly just interested in studying addiction using rodent models. In the past, most of the lab's work was focused on exploring how nicotine can shape learning and memory processes relevant to addiction. But today we really have a variety of projects studying the neural and genetic substrates influencing and affected by nicotine and alcohol use. So to do this research, we need desk space, wet lab, and a mouse facility, and all of these are housed in the Health and Human Development Building, which I have pictured here. And so I was interested in my current laboratory for really the same reasons I was interested in MCIBS. Our lab has an integrative approach using behavioral, cellular, molecular, and genetic techniques to study drug use and addiction. Behavioral assays are really critical to our work. So there's a mouse facility actually across the hall from our wet lab and desk space. We share the facility with only one other lab right now, and we have multiple behavioral suites that are private for our lab where we can do mouse work. And these are Skinner boxes pictured here that we use for our learning tests. Our wet lab is a private space across the hall that we really only share with people in our lab. And we also have a private microscopy suite just one floor above us. So really, most of the resources that we need for day-to-day research are all located in the same place. And then in addition to the lab space, there are a number of core facilities that support our research. So my primary dissertation research question is how genetic differences can modulate alcohol's effects on hippocampus-dependent learning. So to do that, I'm sequencing hippocampal RNA after giving learning and alcohol treatments to mice, but I don't have to do that by myself. So I'm collaborating with the Genomics Core facility who will run the actual RNA sequencing. And then the Bioinformatics Consulting Center, which I don't have shown here, will help me with the analysis. I've also been supported by the Biomarker Core facility, which has helped me just, for example, test blood alcohol concentrations for mouse subjects, and the Metabolomics Core facility, who's doing liquid chromatography and mass spec to help me understand strain differences in nicotine metabolites in blood samples. So finally, there are a number of funding resources available at Penn State for graduate students, but I'm funded by an NIH T32 fellowship in physiological adaptations to stress. And so the main idea is that they'll pay my stipend for 2 years, and in return, I'll take a handful of classes relevant to stress, but also professional development. And so I'll stop here, but these are just some of the resources that have helped support my research at Penn State. Thanks.
Hi, my name is Richard Shen. I am a second-year MCIBS student working with Dr. Melissa Ross' lab. I have a picture of us here just before COVID, and I am sitting here second to the right. In Ross' lab, we are interested in the cell biology of neuron using the model organism Drosophila. Current research topic includes neural response to injury and the cellular basis of neural polarity. The one I'm going to talk about today falls under the cellular basis of neural polarity, which is microtubule dynamics. Microtubules are cytoskeletons that provide shape and structure for the cell. In neuron, microtubule also act as a highway for signaling cargo transportation. I have a figure showing that here, where you can see cargo being moved by motor proteins that works on microtubule like kinesin and dynein. And this is very important in response to events like development, injury, and regeneration. Therefore, needless to say, it is very important to gain deep understanding about microtubule dynamics. So the subject I'm looking at here is severin protein. They are considered as one of the most important regulators for microtubule. As their name suggests, severin protein cuts microtubule, but depends on the cutting site, microtubule can either go shrink down or grow into two. So the hypothesis model I'm currently working on right now is to see if severin protein cut near the minus end where the microtubule is being capped by microtubule nucleation machinery named gamma-tubular ring complex. This release microtubule minus ends from the nucleation machinery and allowed to be bind by minus end binding protein labeled in blue here. An example of that will be petroleum, and this will allows microtubule minus end growth. Also, there are multiple severin proteins that's being identified in Drosophila, things like catenin, spastin, and phygetin, but which one of them are involved in this process is also unclear. The way we tackle this question in the Rolls lab is by imaging live Drosophila larvae with the different genetic background, in this case, such as severin protein knockdown through RNAi and the various severin protein knockout mutant. We track microtubule using fluorescently labeled microtubule end binding protein EB1 through cutting edge microscopy technique like confocal microscopy. I have a picture of here showing one of our scope LSM 800 inverted. I also have a video here of labeled EB1, which we call EB comets, move through the neuron. And by quantifying the number of these comets, it gave us some idea about microtubule dynamics in the neuron. Here, where increase in EB comets number translates to increase in microtubule dynamics, possibly due to the increase in several events, and the decrease in comet numbers possibly is due to decrease in several events. And this is all I have today. Thank you.
Hello, everyone. My name is James, and I am a second-year MCIBS student in the lab of Scott Linder, located on the second floor of MSc. Our lab focuses on using molecular parasitology and structural biology to understand the malaria parasite so we can identify and exploit differences in the parasite's development and weaknesses. In general, we study the species of Plasmodium, which is the parasite that is the causative agent of the disease malaria. Plasmodium falciparum is one species that infects and causes human malarias, and we are able to readily culture this parasite in human blood in the lab. However, much of the lab focuses on rodent malaria species, such as Plasmodium uelii, in order to better study and understand the parasite stages that are involved in transmission, both from host to mosquito and from mosquito to host. The parasite needs to move between host and vector to transmit, and the parasite exists in a variety of stages during its life cycle, moving between host and mosquito. The majority of stages exist in blood as the parasite infects red blood cells. Some of these stages express specialized ribosomes, and my thesis project has the goal of isolating these specialized ribosomes in order to better study its structure, RNA and protein association, and potentially identify the specialized function of it. The Linder Lab utilizes a variety of workspaces for our projects, the first of which is a computational bay area. Many researchers in MSc are given a workstation in these bays as they exist on one half of the second floor of the building. Here is where I'll work on project planning, literature reading, data analysis, and can collaborate or socialize with peers during non-COVID times. We also have wet bench bays where experimentation is performed and communication with lab mates occurs. My lab also has a tissue culture room where I personally culture Plasmodium falciparum in human blood, but the hood is available for others to culture different cell lines as well. We also use this room to prep nearly everything that requires sterile technique. The Linder Lab also has a room in the MSc vivarium to hold and store our mice. The vivarium provides a space for this, also infecting mice with malaria, and a surgical procedure space to perform tasks such as cardiac punctures in order to collect mouse blood and parasites. I also wanted to show a couple pictures from our insectary, where we grow and maintain a mosquito colony for transmission experiments. The insectary is currently located off campus, so we shuttle infected mice in vehicles over for transmission. However, there is an insectary in the works being made within MSC. This space allows us to perform transmission experiments and dissect parasites out of mosquitoes, although dissection often takes place at our wet bench spaces in MSC as well. Lastly, I threw up a bit of information about our microscopy core within MSC, specifically use of the fluorescence and confocal scopes. There are a range of confocal microscopes available to use through the core, as well as 2 super resolution microscopes that are useful for publication quality high resolution images. I also just wanted to list a few collaborations that our lab partakes in. Within Penn State, our lab frequently collaborates with the Haffenstein Lab for structural biology with cryo-EM, and has started doing the same with the lab of Jean-Paul Armache. We often collaborate with the Linus Lab, another malaria lab at Penn State, and even have group meetings with them. We also see collaboration with Esteban Albert for most of our bioinformatics work. Collaborations are not limited to being at PSU though. I am currently on another project collaborating with the Dvorin Lab located at Harvard University. Our lab has many affiliations within the school, including MCIBS/the HUC, Biochemistry and Molecular Biology, or BMB, the Center for Malaria Research, the Center for Infectious Disease Dynamics, and the Center for Eukaryotic Gene Regulation. Thank you so much for listening, and I hope you have a great weekend.
Hi all, and welcome to MCIBS Recruitment virtual style. My name is Liam Kaler, and I'm a second-year graduate student in the Kelly Lab. In the Kelly Lab, we study cancer biology through the lens of structural biology. By using techniques such as transmission electron microscopy and cryotransmission electron microscopy, we aim to determine structural differences between wild-type and mutant proteins, as well as how those structural differences contribute to functional differences. Our lab is located on the 5th floor of the Chemical and Biomedical Engineering building, pictured here. This building is beautiful, and it was just completed in early 2019. Pictured here, you can see our PI, Dr. Deborah Kelly, as well as a current graduate student and a postdoc, as well as an undergraduate student who has since graduated. This is me, as I'm sure you have all figured out. So let's take a closer look inside the building. In the top left here, you can see one of the Chemical and Biomedical Engineering building's main lecture halls. The engineering department runs a weekly seminar where they have guest speakers from various fields of engineering come in and speak with us. Many of the departments at Penn State run similar seminars, so no matter where you end up joining a lab, if you join Penn State, you'll have plenty of opportunities to learn from others. In the picture on the right, you can see one of our lab bays. Many of the bays in the Chemical and Biomedical Engineering building are organized in this way. On the sides of our bays, we have rooms where we store chemicals, perform tissue culture, or freeze our samples for cryo-electron microscopy. As a cryo-EM lab, we're fortunate enough to have the opportunity to collaborate with many other PIs, both within Penn State as well as across the country and even the globe. Aside from our own research, which centers on breast and pancreatic cancer, collaborating with others gives us the opportunity to contribute to disciplines like virology or microbiology. As our lab is also under the Department of Engineering, we also do some work with chip development for electron microscopy. When it comes to electron microscopy, sample preparation is the biggest barrier to an amazing high-resolution structure. But another big factor that our lab considers is the chip. Now, when I say chip, I'm talking about a computer chip type chip. It's really small and could fit on the tip of your pinky, almost on your pinky fingernail. Our lab considers what kinds of materials are going to be most and least reactive to the energy of the electron beam coming from the microscope, with the goal of minimizing that reactivity. A reactive chip could vibrate, which is a substantial problem when you're looking at small proteins at 90,000 times magnification. On the bottom left, you can see some of our lab space. As a cryo-EM lab, we take images with the microscope, and then we have to process them using really high-powered computers. So we have a designated computer space in which we can do that in our building. As I'm sure some of you have heard or will hear later, the HUC Life Sciences has a set of cores where integral technologies are kept together. For example, there's a genomics core where they do sequencing, a proteomics and mass spectrometry core, and of course, a microscopy core, among others. As our lab deals with electron microscopy, I'll be giving you a brief introduction to the electron microscopes in our core. Housed inside the Millennium Science Complex pictured here, the electron microscopes are actually underneath of this garden. By placing the garden on top, the architects of the building are able to minimize foot traffic above the core and therefore minimize vibrations within the microscope, the importance of which cannot be overstated, as I shared before. I've worked with some shaky samples before, and imaging a shaky sample is like trying to get an image of a running dog. It's possible, but it's not ideal, so we try to minimize those kinds of vibrations in any way that we can. The microscope facility, which is a joint venture between the Huck Life Sciences and the Materials Research Institute, is professionally staffed. Shout out to Jen Gray and Carol Bader. This is a great benefit to us as researchers. Not only do we get professional training, which can make a huge difference, there is always someone around who knows how to help if you encounter a problem. As you can see in the top left picture, there are also some flow cytometry machines inside this facility. However, I have not personally gotten the chance to use these in my own research, and I won't be talking about them here. Pictured below are two of the microscopes that our lab uses most frequently, the Talos F200C and the Titan Krios G3. The Talos is a wonderful manually operated scope where our lab can go and collect data on our samples, while the Krios is a fully automated machine which will take images for you. Each has its benefits, but our lab has gotten high resolution structures from both instruments, There are many other electron microscopes which I was unfortunately unable to image for this presentation, but the Huck Life Sciences page has links to the core facilities where you can closer examine the available instruments within those facilities. With that, I'd like to sign off now and keep you guys moving. I just want to leave you with a few notes. Firstly, when entering grad school or picking a lab, I think it's really important to keep an open mind. When I came to Penn State, I did not have a background in cancer biology, and so I would never have imagined I would have ended up in a cancer biology lab. But it ended up being the perfect fit for me, so I really urge you to keep an open mind and try new things if that's what you're interested in. I'd also like to say that if you want to learn more about cryo-EM, I recommend you check out the Symbiotic podcast, season 1, episode 7, titled Cryo-Electron Microscopy and the Resolution Revolution. This podcast is run by the Huck's very own Cole Hans and features my PI, Deborah Kelly, as well as a research professor, Cameron Verano, and a microscopy core staff member, Jen Gray. With that, that's all I have for you. And if you guys have any questions, feel free to email me at the email below, ltk5073@psu.edu. Thank you.
We hope you enjoyed listening to some of the MCIBS students talk about their research in this series. Now we have two former MCIBS graduate students who are currently pursuing their postdoc after they did their PhDs here.
I think that what I'm most excited about in terms of my research is leading the forefront of the new information that's coming out in terms of cell biology. So in neurons particularly, there's a controversy as to how their microtubules are organized inside their cells, and the work that I do is particularly interested in figuring out how they're organized, and it is in contrast to sort of the dogma in the field right now. And so rewriting some of that dogma is really, really interesting. So I got into two schools. The other school did not have as wide of a breadth of programs as Penn State had to offer, and I really wanted to explore a wide breadth of the different programs that I could study. So some of the programs that I was interested in were human evolution or ecology, as well as cell biology, and MCIBS really allowed me to explore those disciplines. I think as a Penn State grad student, it's allowed me to grow as a scientist in particular, and give me the opportunities to be able to enable others who are coming in, new students or even people in high school or middle school, to be able to explore science as a possible profession. And so I think that the grad program here has really helped me to explore those options.
feel that I'm really passionate about what I do. I really want to help people that have genetic disorders, even finding a little bit of the answer and to try to understand why do they exhibit the features that they exhibit, to have a treatment, to have that, piece of what they know what their disorder is about. So that's what really brings me here every day to keep doing it. There are different approaches to answer the same question, and it's a very collaborative environment in which all of us work in the same project from multiple approaches. So definitely the interdisciplinary nature of the Hack Institute is what really sold me on coming to Penn State.
We'd like to conclude the series by sharing a video that highlights the interdisciplinary research that takes place in the Huck Institute of Life Sciences.
Interdisciplinary research is trying to look at what the challenges of the day are. What are the big issues that we're looking at, and how do we solve them? You think about things such as diabetes, neurodegenerative diseases, personalized health, growing plants for food and for drugs and things like this. You need multiple disciplines to do that.
We want to really to push the boundaries, and to be able to do this, we need different brains coming from different experiences, different way of thinking. We need to compare and share ideas.
If we want to build a team, then we actually have to bring people with different expertise together.
Talking about One Health.
We're working on foot and mouth measles and Ebola.
A cluster hire can change the course of a university research program by hiring one there, one there, one there, and all of a sudden you have this powerhouse that you can assemble more or less overnight. It's magic, and it's the future.
I come into work every day as if I'm playing in a sandpit. In this building, I can walk next door to meet a statistician. I can walk a few doors further on to meet a sociologist or an anthropologist. The Huck Institutes are really a place for innovation, so they kickstarted Plant Village. 8 years later, we are engaging across an array of individuals and institutions globally. So in a sense, we've made the sandpit global.
We put this wonderful building up, the Millennium Science Complex, which is the material science people and the life science people.
The Huck facilities actually exist in a total of 6 buildings. They range from genomics, proteomics, metabolomics, microscopy.
Corporate partners come in to use our facilities as well, because we have cutting-edge technology, cutting-edge research, and scientists of world-class caliber.
One of the easiest ways to describe the Huck Institutes and its core facilities is in terms of what we don't have. It's a very short list.
Our graduate programs offer the opportunity to work with the world's best experts in a variety of different subjects.
Enzymes have the power to turn Mount Everest into Mount Nittany.
We've got one of the best centers for infectious disease dynamics in the country, perhaps in the world. I'm working on a disease called Peste des Petits Ruminants virus in northern Tanzania. It affects about 80% of the world's small global sheep and goat population. We're just really interested in making strides preparing for the upcoming eradication efforts for this disease.
The first impression was it's really friendly. It has really diverse research topics, and it encourages students to explore all the potential options by doing lab rotations during our first year. It was a really awesome platform for graduate training.
I work hard, but I also find balance in my life. I enjoy going on hikes. There's beautiful mountains and trails around here. I've joined Club Frisbee to stay fit and exercise after work.
I love being a graduate student because I have a family, I have a wife, I have a kid, I have great health coverage, and I get paid. I love what I do because discovering new things That's what really gets me excited when I wake up in the morning, is today I may find something that literally no one in the world knows.
In multi and transdisciplinary teams of scientists, you want people who play well together.
And having all these people together in the same place is really important when you're trying to solve complex, multi-layered problems in global health, epidemiology, or infectious disease. And I don't know another university that puts all that together in one place.
The Huck really enables me to do research at various scales, from the small all the way to the big. They're really preparing me for just about any kind of career broadly, and I'm really thrilled about that.
The technology that we have now means that the limiting factor in terms of what you can do in your research is your own imagination.
Penn State is really rocking. We're doing the most remarkable things here. We've got the instruments, we've got the faculty, we've got the environment to do that. Come here and you will see it. Come and be part of our team.